Can we assess what we do not see? By Dr. Itai Harman. He's senior lecturer and assistant professor in the Robert H. Smith Faculty of Agriculture, Food and Environment, Hebrew University of Jerusalem, Israel. I would take an opportunity to give his very short, brief introduction. He's currently working at the Faculty of Agriculture, Food and Environment. He has done his BA with Geography and Environmental Development. He has completed his MA at Ben Gurion University, Jacob Houston Institute of Desert Research. He has also completed his PhD from Ben Gurion University. He was postdoctoral fellow at Ben Gurion University, Jacob Houston Institute of Desert Research, and he has completed his postdoctoral fellowship at the University of Wisconsin-Madison, Forest and Wildlife Ecology. He is extensively working in the field of precision farming with 30 numbers of research papers, publication and reputed journals, which is totally inclined to different aspects of precision agriculture using spectral sensing and other satellite applications. We are fortunate to have with us Dr. Itai Harman. May I request Dr. Itai for his expert talk and give us some information regarding his work and his experiences, please.
Okay. Thank you very much for the introduction. I hope it's sharing the screen nicely now.
Yeah.
Yes, it's okay.
Yes. Yeah,
nice.
It is
okay. Great. So thank you first of all for the introduction and the invitation to be here, and I'm very happy to do so. I've heard we have something like 1,600 people, which is very nice. That's it. So let's start. The title for the talk today is Crop Traits Estimation by Spectral Sensing. Can we assess what we do not see? The idea is to talk about ability to assess plant traits, and we can do it with a visible range, and we can do it beyond it, and we can look at traits that we can actually see, like colors or like pigment content, or we can look at traits that we cannot see, which can be like if we measure the plant today, what is going to be the yield in a month from now? Or if we look at the plant today to learn about the water potential, which is the pressure of water in the leaf and stuff like this. This is the idea. And let's start. So first of all, So first of all, I would like to present our lab. We sit in the Hebrew University of Jerusalem, in the city of Rehovot, in the Faculty of Agriculture and Plant Science and Genetics in Agriculture Institute. Our lab is a plant sensing laboratory. We can see a group of people. One of them here in the white shirt and the triangle is also from India, is Maitri Asahu. And we are at this time, or in general, we are relatively young, only 3 years old, and our focus is at trait estimation, hyperspectral data analysis, high-throughput phenotyping, and precision agriculture. We work with UAVs, we work with satellite imagery, we also work indoors with cameras, we work with plants themselves, not only the spectral data, but also collecting the traits. And we collaborate with people here in our institute that are agronomists and genetics and people that understand physiology of plants. And the idea is that we work in a team with many people, because I think we'll talk about it also, but I think working alone in this time, it's a bit strange. We are working as teams to have better results, a better understanding of what's happening. So this is it. Usually, sometimes it's in the end, it doesn't get enough attention, but I would like to appreciate many people that we are working with and have worked and are working now, from Israel, from India, from United States, from other places. And this is a partial list of the research that we're just presenting today. So our research will be, in general, it will be the motivation for plant trait estimation, just to understand why, a bit of background of a little bit of remote sensing, a little bit of precision agriculture, a little bit of breeding. Then we'll focus to vegetation spectral reflectance to understand it, why it looks like it is, and then we'll see the examples like applications of using it. This is the plan that I've been planning for, and to the best of my understanding, I have something like 45 minutes or so. So the motivation is food. I guess everybody knows this. This is chickpea. This is sesame. If you put them together, you get a nice dish of hummus. And you can eat it with a fork, or you can eat it with a pita bread made out of wheat. So this is this is a motivation. As we know, the population of the world is growing. The good news is that the rate of growth is decreased, but we are still going to be more and more and more, and most of us like to eat. And we need to produce food. This is a map done by the UN. It is talking about what the productivity of land will be in 30 years from now. Red, it means the bad color. Green, in this case, green, the good. It means where is it red? In places that it's red, it means that the productivity of the land will be reduced. So we see the global way of looking at it. For me, it's strange to see that there is a border, let's say, between Mongolia and Russia. And so one leg in 1 foot stands in Mongolia, 1 foot in Russia. In one side, it's deteriorating, and the other side, it's the productivity is growing. So I think it's also related to the regime and politics and stuff like that. But in general, I think we should pay attention and make sure that the agriculture that we do is sustainable and will allow us to keep on doing it for many years. This is a lesson that I take from this map. What about the future? The future, there are all kinds of profits, but who knows? There is a book written by Nassim Nicholas Taleb. He's a guy from Lebanon. Most of his life is in the United States. He was working in doing a statistical analysis in economics, and he wrote a few books. The most famous one is A Black Swan. A black swan event for him is an event that is unpredictable outlier. It has an extreme impact, and when you look back, you can say, I should have thought about this one. And I guess all of us has one example in mind, and I guess that for most of us, it's this one. But there are other examples, like the invention of penicillin, less than 100 years ago, and about this time in the 30s, the Dust Bowl in the United States. A little bit around 90 years back, people were hungry in the United States. There was a period of few years of drought, and crops could not have been grown on rain summer, on summer rains. So it was a big problem. And some people say that the computers and Internet of Things and connection between computers and agriculture is a new black swan. So the idea, if we need more food and we want to produce, and we want to produce more, and we need more food, the idea is to increase the yield per unit of land area. So there are two main ways to do it that are also already done for several decades. One is agronomical practices, to know when to sow the plants, to know how to fertilize, how to irrigate, when to do it. All the agronomical activities that makes the plant grow, that support the plant growth. And this is usually falling under the wings of precision agriculture, because on one hand, you can look at the entire field and treat it the same, or you go for specific regions in the field and treat them specifically. But the word precision in precision agriculture, it's not only for location, it's also for the right treatment, precision in the type of treatment, the right time to do it, the right place, and the right manner. So you can add nitrogen, but you can throw it on the canopy, or you can put it in the ground, or find the best way to irrigate. So it's not only the location, it's also other stuff. and to do it in commercial fields and to find the viability in the field, on the land, on the soil level on one hand, and on the canopy level, on the vegetation level on the other hand, there is a need for a lot of information. As we can see here, there are layers of information: soil type, yield maps, maps of weeds from previous year, diseases, and more. And there are huge amounts of data to be analyzed. And we can see that today there are even manufacturing tractors without drivers. There are GPS, and the tractors can drive as easy with nobody behind the wheels. So this is one direction, is agronomical practices. The other direction is breeding. So you can breed for a wheat cultivar that will be more resistant to this certain disease, or have the longer roots, so it will get to water when other cultivars cannot. So it's more resistant to drought and other things. The idea that for the few decades, these two concepts of agronomical practices and breeding are going hand by hand, and they are going hand by hand. So I'll mute my phone. They are going hand by hand, and each of them gives some kind of contribution, and the combination is bringing us to what we have, because for the several decades we have an increase in yield, I think in almost any crop, in yield per unit area, per land area. So this is breeding. The differences between precision agriculture is talking about commercial agriculture. So you have fields and you need to treat them. Breeding is more in a breeding projects. So you can find the best cultivar for a certain region or for a certain disease, or check the traits of cultivars that are being developed. So it's a completely different scale of work. You can see, like in comparison to commercial fields, you can see a plot for experimental for breeding, and we'll talk about that later. So I guess for precision agriculture, the most common tools are satellites and sometimes sensors on tractors that go in the field anyway. And for breeding, it is more likely to be using UAVs, but it's not a rule. Things are flexible, pending the aim. So if you want to do high throughput phenotyping and collect a lot of data, this is a system made by a company called PSI in Czech Republic. It works indoors. It can shift the pots from place to place, and you can measure them in chambers. Each chamber has its own camera or sensor, and you can get tons of information: hyperspectral, multispectral, RGB, fluorescence, thermal, and more. And similar system by Lemnatec from Germany, they build the bridge, and you can do it in the field. Also PSI they can both do the same. In Zurich, in the universities, they use sensors running on cables, but now they use more UAVs. There are all kinds of tractors or similar to tractors that can drive in the field, and you can put on them all kinds of sensors, and you can collect a lot of data. You can do it with the robot, like they did in Australia and Sydney, and you can do it you can do it with bicycles and put the devices that you want also there. But the most common, or most fashionable now, is using UAVs, and it makes sense. It's without boots on the ground, but on the other hand, you need to find people to operate it and to run it and to analyze and etc. That you'll do anyway with data. So this is what we have. If we talk again about precision agriculture, I think that most of us, when we think about precision agriculture, we look at the two bottom pictures. Like the UAVs talking to the cameras, talking to the robots, talking to the combines, and there is like a command room. All the data is coming in, so you can control everything. And this is the idea. But precision agriculture is also what we see here. Somewhere in Eastern Europe, somebody has found a way. Instead of walking in the cucumber fields and trampling over the plants, you can lie down and pick them while the tractor is driving. So you increase productivity, you reduce the damage to the plants, and this is precision agriculture as defined by the ISPA, International Society of Precision Agriculture. Every word here counts. People have been, like scholars have been discussing about this for, I think, almost a year. So every word here counts. So precision agriculture in general, this is what we see. We have lots of data of input. A tractor driving, it can have like a sensor that will learn about the soil, all kind of sensors in the soil about the moisture. You can learn beside moisture, you can get pH, you can get microbiome, you can get nutrients in the soil. You can use all kind of sensors by tractor, you can use UAVs, you can use satellites. You have genetic information. All of it is going to the cloud. Eventually you get a map, and then the tractor can drive and fertilize or apply herbicides as needed or pesticides as needed. So the idea, there are tons of data. And if we want to do it right, the suggestion, if we're doing so, first of all, we need to collect the data, and the data should be collected in the right manner. It should be collected in a good way. And beyond that, the data should be analyzed in a good way, should be pre-processed and analyzed, and eventually you get conclusions. This is for breeding. So you find this cultivar is less or more susceptible to this or that disease or abiotic stress. And if it's not done with caution and smartly, or with enough people that understand what they do, because on one hand we can take pile of data and give it to a computer science scholar. He will run it or she will run it, and you'll get a nice graph. But the meaning is less important. For example, for here, we can see on the x-axis, it's fresh lemons imported to the US from Mexico, and the y-axis is total US highway fatality rate. So the R-square is very high. The arrow looks like very small, so it makes sense. If United States wants to reduce the highway fatality, you should keep on importing more and more lemons from Mexico. The R-square is very high, so it makes sense. So if we want to prevent graphs like that, we need to do the analysis with people that understand data, with people that know how to collect the data, with people that understand the plants. And this is the reason that we need multidisciplinary teams. So if we want to go for the next level for precision agriculture, it's not enough to assess the traits, but it's also there is important for giving something to the farmer or to the grower. For example, the spatial decision support system. So if there is a field, the farmer can get a recommendation or get a map, and he will decide or she will decide what to do. But the idea is that to give the farmer with information, give them the information to decide how to treat the field and what to do it, and probably don't treat the entire field as one unit, but look at it as what's known as management zones, parts of the field that are treated differently. And the last piece is a variable rate application. Let's say that we have found that this tree needs to drink more water than the tree next to it. How can it be done? So it's not always easy to do, and in some cases it's very complicated. So this is the levels of data analysis and what can be done in the direction of breeding and precision agriculture. For the background, the sun that we have emitting in what we see here in yellow, the x-axis is wavelengths, the y-axis is energy radiation in irradiance units. The visible part is this one in the middle, from 400 to 700, and most of the energy is there, like about 45% of the energy is there. The yellow is whatever is the sun is emitting. The orange here is whatever passes through the atmosphere and gets to the soil level on Earth. It gets to us on our plants, on our cars, on our highways, on our buildings, on our cows, wherever. And if we look here, we can see that ozone has a big effect. We can see here an absorption by oxygen. We can see absorptions by water and by CO2. So the atmosphere is filtering a lot of the radiation that is coming in. And it's probably not surprising that we as humans and plants are using the visible part for us, for visual, to know where we are, and plants are using this area for energy, doing photosynthesis. So if we have a sensor the sun is emitting the radiation, it hits the plant, and the plant is reflecting something back. And this thing we measure with our phone or with the sensor. So we measure the reflectance from the plant. And this radiation goes in the sensor. These photons that comes from the sun reflected from the plant and going to the sensor, probably silicon, and then electrons are coming out, filling wells, and then we have a little number in our pixel that says this is 72, which is related to the amount of photons that was hitting the sensor. And if we're using cellular phones, we have super pixels, like 4 pixels will create 1. So with little filters, less a few microns in size. And we have blue, green, and red color, and we have information. We can use also with much sensor with much more information, like hyperspectra. I've seen on the plan that there was another previous lecture talking about hyperspectral remote sensing in precision agriculture. So I might be saying things that were already mentioned, so I'll not go deep to that. If we look at the research way of looking at things, like the prospect of research, we want to do some kind of analysis. So first of all, we need to have a research question, some kind of question. And when we have it, we start selecting the tools. Let's say the spatial resolution. It can be a pixel of millimeter or pixel of 30 meters or 500 meters, depending which satellite you use. Spectral can be 1 band or more, as mentioned, and temporal can be measurement like continuously every second for 7 years, or it can be measured one time, or it can be measured once a week. And there are all kinds of scales and devices and ways of looking at things. It's important to remember that we can't always get what we want. It means that we cannot get like a millimeter pixel with a hyperspectral resolution measured every 5 times a day, covering the entire globe. It will not happen soon. But the good news is that we're getting there. We're getting there, and we're having more and more data, which we need to put in mind that sometimes too much data is too much, and not all the data is needed. And we need to find what is relevant for our aim. And this is the way to select the tools to analyze, to use to answer our research question. Here is just a slide to show the differences between RGB, which is like 3 points. We don't know what goes on in between. For us, it's just a green line. If we have like multispectral, we have more points. We still don't know what goes on in between. This is a red line. And if we have hyperspectral, you have many points on this line. It's defining the reflectance very nicely. And with the high resolution, we can see every little trend in this line. We can see even this little peak that is related to fluorescence of the plant. And it's visible because there is an absorption of oxygen, if you remember a few slides ago. So this ability to detect fluorescence of plant is important, and the Europeans are about to launch a satellite in the coming year or two for that, and a lot of studies are done for sun-induced fluorescence. In our lab, we are less doing it. We've done something in the past, but we're less doing it and focusing on other stuff. So what we see here is on the upper right is the blue one is reflectance of water. The brown one is reflectance of soil, and vegetation is the green. It has a unique, distinct shape. And why does it look like that? In the visible part, as we see here, most of the radiation, very little radiation is reflected. Very little radiation is reflected. It means that most of the radiation is absorbed, and is absorbed for photosynthetic process to get the energy for the plant to produce. In this region, the near infrared, half of the radiation is reflected, and the level of reflectance is set by the amount of leaf levels. If we have more leaves on layers of leaves, more biomass, vegetative biomass, will have a higher reflectance. And as we go for the shortwave infrared, the effect of water is bigger. This is an absorption of water, absorption of liquid water, and the water is affecting very much, but we can see that water has effect also here, and biomass is also affecting these parts. So in general, if we look at the hyperspectral or spectral curve, the information that is reflected from the plant contains in it all the traits of the plant. It means how many leaves there are, the inner structure of the leaf, the chemical properties like pigments, nutrients, if there is a wax on the leaf or not, and many more things, water content, other metabolites content, and etc. So it looks like the hyperspectral is a lot of data, and we need to learn how to get out some information from it. Just for example, if we look at chlorophyll, we see that the red color in the legend shows less chlorophyll, blue shows more, and we see here a reflectance curve. So we can learn from that if the reflectance is high, it means that there is less chlorophyll in these regions. And we can see also this area that is known as the red edge, that if the reflectance is this line is moving to the left side, like towards the blue wavelengths, we're losing the plant is also with less chlorophyll. So this is like relation of spectral data directly to a trait of the plant. Chlorophyll we can actually see because we will see it by color and we can guess. But on the other hand, there are plants that we look at them and we'll see them as purple, but the amount of chlorophyll inside is the same. Or we look at them and we see the leaves are white, or parts of the leaves are white, but if we extract the chlorophyll, we'll see it has the same amount of the other part of the leaf, which is green. So it's because of air gaps and stuff like that. But in general, chlorophyll is something that is visual, but not in all cases. So this was also some kind of background for us to look and understand about the spectral reflectance of vegetation. So I want to go for an example. If we look at chickpea, the chickpea plant, we see that India is a huge producer of chickpea. 65% of the land on earth that is used for chickpea growth is in India. On the other hand, if we look, and Israel is not even shown here because we are too little. And if we look at countries and look at yield, but not the just amount of yield, but yield per hectare, tons per hectare, yield per land area. And we see that the little country known as Israel is at the first place with 3 tons per hectare yield, while Australia and other countries, and including India, are with lesser yield, like 3rd. The reason for that is irrigation. In many places in the world, the chickpea is not irrigated. In Israel, in most cases, it is irrigated, and this is increasing the yield. But there is always a but. But if we look at the leaf cycle of chickpea, in Israel at least, it is planted at winter time. And then the seeds are naturally irrigated by rain. And after the rain, the farmer needs to decide when to start and irrigate. Because on one hand, if the minute the rains are over, you start and irrigate, what will happen? The plant will not have any stress and it will keep on growing and flowering will be delayed. And then you will have a very big plant with a very little number of pods of seeds. And then the harvest, the yield will be lesser. On the other hand, if you wait too much until you start and irrigate, you save water. You wait too much, the plant will start terminal stress. It means that you will reduce just very little. It will flower. At the stress, it will flower. And then it will start putting all its energy to the seeds that is already there and will not produce new flowers. So we want to be in the sweet spot. We want to wait enough time for the plant to severe a little bit of stress to start flowering. On the other hand, give it water so it will not go to terminal stress. Give it water so the plant will be good, will have flowers, and then more flowers, and then more flowers, and then more flowers. So we'll have more and more layers of pods. As you can see here, 1 layer, 2 layer, 3rd layer. and there are layers of pods, leaves and flowers, leaves and flowers. This is the idea. So the question, the big question is when to start and irrigate. And to do so, farmers in Israel, what they do, they look for the water potential. So they pick a leaf, put it in the pressure chamber. It's a chamber that only the petiole of the leaf comes out. And then you increase the pressure on the leaf itself by gas, like air. You push air in it. And then the minute you see water coming out of the petiole, you know the water potential. And there are numbers. You want to be at this water potential. If this is the case, you need to irrigate. This is the idea. So the plant has been under enough stress, but not too much. And then this is the time to irrigate. But if you do so, you have a big field. Each measurement like this takes, 10, 15 minutes. You cannot cover the entire field. So here comes remote sensing. We can do something like that. So what we have done, we have used 3 tools. One is a satellite. It's an Israeli-French satellite named Venus. It's not commercial. It's for research purposes. Excuse me. It's for research purposes. And it has 11 spectral bands. And it comes the revisit time is 2 days. And the pixel size is 5 meters. It's quite unique. It's similar by traits to Sentinel, but it has improvements. On the other hand, Sentinel is covering the entire globe, but Venus has specific sites around. So we used the satellite. We used also a UAV-mounted camera, a 5 bands camera, and we use also a hyperspectral ground-level sensor. We used 2 of them. One was facing a white panel, as you can see here, to get the reflectance from the sun. And the other one was going around in the field, measuring the plants. So we can correct for the sun, and we'll talk about it just now. Because let's say that I'm looking at my hand, and I'm looking at it now. I'll take a picture. The pixel will show a value, of 200. But now I will shut down the light. I will take a picture in a different illumination. In a second later, the value in the pixel will not be 200, it will be 70. It's the same hand. So we need to work on relative terms, like the idea is to measure the radiation that is reflected from the target, but in relation to the incoming radiation. So we want to know how much we measured in our device and divide it by the incoming radiation from the sun. And then we can get the right value. The example here on the left, we can see the black line is the sun, the red line is grass, as we see here. This is under sunny conditions. They're almost the same target. This is under clouds. We can see the sun is showing less on the ground and the plant is reflecting less. If we divide the red by the black, or here the blue by the black, we get very similar results. It's not as simple as is. We talk about the IT here stands for integration time. We reduce the dark current, etc. And for the field of view, we cover for it. You can look at it in a paper that we had in 2019, papers in the Journal of Sensors. That's it. And if somebody have questions, you're always welcome to contact via Rucha that invited me for here. This is how we measured. If we look at data, result that we have from this experiment, we can see here, this is day 74, day after emergence. This is, all the plants are the same. There are different plots in the field, but all plants are irrigated evenly. We can see the spectral reflectance for 6 treatments, 0 water, 50% water, up to 140% water, which is more than what the farmer would do. But this is before the irrigation treatment has begun. So all the plants are almost the same, all the traits. Here we look 20 days, 21 days after the differential irrigation began. So parts of the field has not been irrigated at all. Parts of the field have been irrigated by 50% of what is recommended to be irrigated. And parts have been like 100% irrigated. This is like the control part in this treatment. And 2 treatments have been irrigated too much, have been over-irrigated. We can see the colors here. We can see the The plants that are yellow, these are the plants with zero irrigation for 3 weeks. And we can see the reflectance. We can see, look, check the colors, green, yellow, sorry, black, yellow, oh, it's green, yellow, another green, blue, and you can see that the reflectance here in the pigments and the water is the same, and it is ordered by chronological, by the different treatments. And we can see also in the near, but it's going the other way around. So plant that has been irrigated less, it has less layers of leaves, and then it reflects less. A plant that was fully irrigated has more layers of leaf, and then it reflects more. This is for here. For the pigment part, if a plant is fully irrigated, it has a lot of pigments and they absorb the radiation. The reflectance is lesser. A plant that is less irrigated, it has less pigment, it is reflecting more. This is what we see here. So the experiment was successful by applying the treatments. We can see here also, this is a thermal imagery using a thermal camera by Dr. David Bonfil. He's a chickpea expert and a precision agriculture expert, and also in other things. And what we see here is just the thermal to see and see the temperature. We can see plots with higher temperature, it's 32 degrees. These are not irrigated. If we go one back, you can see this area. And we can see it here now. It's this area. It's the same. It's less irrigated. The temperature is high. 32 instead of 30. If we look here at this scatter plot, we talk about the measured leaf water potential. We talked about picking a leaf, putting in a chamber, applying pressure, and looking when the water is coming out. We can see the relation between the measured and the predicted water potential using spectral data, as we've seen before. The spectral data that we've seen before, we've developed models, and we came to R square and root mean square error of prediction to estimate leaf water potential. This is a trait we cannot see. We cannot see the water potential. It's a physical unit. Another example, this is like, this was leaf water potential from the ground, from ASD, on the ground in 4 sites. 2 seasons in one farm and 2 commercial fields. What we see here, it's still water potential, but only from the satellite. This is data from the satellite in the 2 commercial fields in 2019 and 2020. And you can see here that we can keep a similar quality of ratio. Even here, the R square is a bit bigger, but the error is also growing. And we can see it's possible to assess leaf water potential also from space. We are now at these days writing the paper to publish it. This is a field, one of the 2 fields. This is Tzion, the one from 2020. In the center, we see here the treatments with 100, 120, 140. Here it's a 50% irrigation, and here it's 75. And this is the rest of the commercial field. And we can see that if we apply our model for leaf water potential, we can see it. In the most irrigated ones, we can see a nice water potential. In the 50, we can see lesser. The water potential is increased. It means the plants are drier. And we can see that it also goes for the commercial field. Another something to look at is the yield prediction. This is 1 year. This is the second year. The first year, we didn't have the zero irrigation treatment. So it's only 50, 75. You can see we can get to above 5. You can get almost 6 if we look at the averages. And at the next year, we do not go over 4. At this year, at the second year, we have a 1 week of heat wave. It was like 6-7 days of more than 40 degrees Celsius. So the idea was people have made, like Asaf and we make the plants stay alive, or just to keep them alive, but the few layers, 1 layer or 2 layers of flowering was reduced, and the plants, the whole development of the plant was stopped in some cases. So we can see the reduction in the yield. If we look in the x-axis, this measured yield grain, and this is predicted. We use spectral data from a certain date to predict what the yield is going to be in harvest. And If we did it for 1 year and for another, we can see the days after emergence, and we can see the x-axis, it's the actual error of the yield. So we look for the smallest error, and we find it's in day about 90 in both seasons, and we put 2 seasons here, and this is a chart that we've got. So we're also able to predict yield from this day forward. It doesn't mean that it will work anytime it is working, but this is what we got for 2 seasons. We've already done the 3rd season, and it looks very similar. So it's for 3 seasons of analysis, so it looks like going to be a good thing. This was chickpea. Now we're going for sesame, as we said, on our way to build the dish. It's a sesame plant. You can use it to make a tahini sauce, or you can use all kind of candy. And here we're lucky enough to find a picture with a bee flying next to it. So this is sesame. We had an experiment for several plots, and we can see that there is a nitrogen gradient. From here, less nitrogen, here more nitrogen. You can see it's affecting in 2 ways. One way is color. We can see that the color is different, less nitrogen, less chlorophyll. So we can see it. On the other hand, we've also developed a 3D models. Like if you take a camera, it's flying, and you have the same spot, take image from several directions, you can estimate its relative elevation from the ground. Like if you cover one eye, you lose the depth concept. If you can look at something from few directions, you can find its relative elevation. And this is what was done, and we can see the blue lines are like isolines, like topography. All the orange line are less than 120 centimeters. Whatever is blue is 180 centimeters and above. And you can see that with low nitrogen, most of the plot is less than 180. With medium nitrogen, more of it, and with high nitrogen, more of it is higher. So this might be an effect of the nitrogen. The entire, it's not moving. Okay. Looks like it decided to stop. Restart the program. Sorry, but it looks like the PowerPoint has crashed. Sorry for that. Screen sharing is stopped. I'll upload it again and keep on. I'll go here. Okay. And start and do that and share, and we are here. Share. Is it okay now? I hope it's okay.
Yeah, it's visible.
It's okay. Sorry for that. So what we can see here is the experiment. We can see other parts of it. We've been looking before on this part. We can see that this our UAV with hyperspectral camera. It's a line scanner. It takes a line after line after line, and it flies. So sometimes it's shaking a little bit. This is the raw data. We can see it's not fully straight. The UAV was not flying completely straight. You can see that there are some bends. If we look at the top here for correcting for sun radiation, we can see that the reflectance is high. As expected, it looks like reflectance from the sun. If we look at vegetation, it looks like from vegetation. And if we divide one by the other and correct some geometry, this is what we get. This is a reflectance of plant from our hyperspectral UAV-borne camera. And this is when we put the lines back together. It's not complete, it's not perfect. We can see here, this area is a bit tricky. Usually when we fly, we do it 2 times. So we'll get one good. And we try and check in the field before we go to the office. So this is an example to show that nothing is perfect. You can see the size of the field based on the cars here. And you can see the experiment that we had. It was something like 30 plots under different irrigations in one cultivar. We did also some measurements in contact with 5 different sesame cultivars. And we can see the reflectance from the leaf, the entire reflectance of the leaf, and we can see that different cultivars behave differently. And it's not just the intensity, some is upper here, lower here, and you see that it is changing, and we can see the differences. And if you want to calculate for chlorophyll, there is a very known index called TGI, and it actually calculates the area of this triangle. This point is defined by the two axes. This is X, and this is Y, and you use the X and Y here to calculate the area of the triangle. And based on that, we can see for each day, day 33, day 34, day 47, 48, 61, 62, we can see that between the dates, there are no big differences. But if we go along the season, we can see that there are some differences, but in general, each of the cultivars is maintaining its own chlorophyll level, or at least relative chlorophyll level to the other cultivars. It's interesting to see that the chlorophyll is related to the cultivar itself in some cases. So this is nice to see. Another thing, if we look at the same plot, we had two types of treatments. This is an RGB, so it's much clearer. The pixels are smaller, so you can see very much better now. WL, it means water limited. 15, it means low nitrogen. And we have water limited with low and high nitrogen. WW, it's well watered with low nitrogen, and well water with high nitrogen, which is controlled. The idea is to see if we look at day 36, 42, and 52, and DSI, it's like a combination of two bands, one minus the other, divide one plus the other, to have some kind of spectral analysis. We can see that irrigation has started here. Differential irrigation started here. Until this point, all the plots look the same. One day after, the plot looks the same. 10 days later, we can see that there are some differences. The green one, low water and low nitrogen, is very different, is significantly different, A and B, from the control group, from the high water and high nitrogen. But on the other hand, here, the two limited projects, like one with a lot of nitrogen but less water, and another one with a lot of water but less nitrogen, it's hard to separate between them. So if we'll throw in thermal camera, we'll be able to separate between them, because the ones that with water limited will shut down their stomata and they will appear to be hotter. This is what we assume. We haven't done it yet, but we might do when we'll get our thermal camera. So if we look at chlorophyll content, we can see a very nice relation between chlorophyll and the arrows. We can see here a very nice relation between the treatments, between the ability to assess chlorophyll content here. We can see the R square is very high, the arrow is very small. We talk about an arrow is smaller than 3, and the values are 30, 35, 50. So you can see the arrow is very little, and it's very high ability to assess, to estimate the content of chlorophyll. And we can see the small arrow. Another experiment of Sesame, it's a very big, relatively big one. You can see the size of the car here. We have about 1,000 plots in this experiment. We have something like 200 cultivars in here, and this is duplicated 5 times. One here, one here, one here, and one here. So each piece like that is one block. If we zoom in, we can see the plot size. If we zoom in further, we can see our tabs and like georeferencing targets. And if we look carefully, we can see that there is variability in colors. And if we look closer, we can see variability also in other things, in height and in maturity, because here we see a plant that is ready to be harvested. Here there is a plant that is just starting to yellow, and here is a plant that is still green, very green. All plants, all sesame plants have been planted on the same day. One cultivar was finishing his life cycle very fast in comparison to the other two. and there are differences in physiology and morphology. You can see how high they are. So this is paradise for people that do genetics, and this is why this panel of sesame plants was selected by the sesame expert here. It's Professor Tzvi Peleg. And he selected this sesame panel to develop, so we will be working and developing some phenotyping techniques. And this is what we've done. We're also working on with wheat. We've done 1 year experiment with wheat. We can see the plots are UAV. We can see the plots here are smaller, and they're about 500 plots. And the idea here is to combine, because genotype is genetics, and environment is affecting, and management is affecting. So the idea is to combine the effect of the genotype with the environment and with the management and learn. Because if we want to develop or to identify or select for breeds for cultivars that are less affected by a drought or less affected by disease, we need to test them. And one big important part is genetics. This is a genotype. Another part is the environment, because one genotype can have a great yield if it has enough water, but if it's the precipitation is lower, so the yield will be dropping. Another one will have less yield, but it doesn't matter if it has more or less irrigation or precipitation, it will give very similar yield. So you can prefer where to go. And the idea is to select for that. And if you throw in management, is also something that the farmer can do beyond the environment. So it's a combination. Environment can be precipitation and temperature and day length and stuff like this, but management can be more like when to irrigate, when to plant. So this combination of genotype, environment, and management is very important. And the big project that Zvika Peleg is leading with wheat is also, we're doing similar stuff. Just preliminary results for the wheat experiment. We can see the hyperspectral imagery over the RGB, and we see that they fit out nicely. And this is ability to assess leaf area index from the UAV. And this is important for us to look for, to assess how big some kind of the cultivars are, and to look at growth of the plants. Another example for wheat is also assessing chlorophyll pigments. This is total chlorophyll, this is chlorophyll B and chlorophyll A, and this pigments has different roles, and it might be relevant to have both of them. And looks like the PowerPoint is less cooperative. Okay, and now we have here also the arrows, and we can see that the arrows are very small. Just to wrap up, we have in our facility also a rainout shelter that allows, it was just opened, and we are going to have here the first experiment. It's already done. Roy Sadeh is a PhD student. He's working on it, and we're about to plant in less than 2 weeks, and we're going to run it. The idea is that when it's raining, this roof is covering this area. In here, we'll grow plants that will have rain, and in other days, the roof will stand in this part. So this is just parking. Here, it's like drought treatment, and here is like full irrigation treatment, all natural. And the plants will have the same conditions, temperature, day length, soil, everything is the same. The only difference is blocking the rain. In Israel, we have winter rains. So for summer rains, for summer, it's easy to have to check for irrigation, but for winter, we need to have that. And this is the only facility in Israel. It was just built. And that's it. That's it for me. If I would just like to say the two main things I think that I would like to say is one, that there is no one way to do the analysis, but there is but always select for the good way. Make sure that the data that is collected is good, and the analysis makes sense, and check yourself. And we try and do that to make sure that the results that comes out make sense. And it's not like we'll be importing lemons to reduce highway fatality. And this is one thing that I think it's important. And the other thing it's important to say is that remote sensing is good, is very good, with doing a lot of trait assessment and using for decision making and great analysis, but it is not the only solution. There are many other solutions, and remote sensing, I think, will be best used if combined with other disciplines. It's not like that the satellite will work and nobody will go to the field again. People must be in the field and understand the plant and the soil and the diseases and the weather and everything. But remote sensing can give extra and and make things more efficient and allow getting more better decisions. This is the idea. Thank you all for listening. If somebody wants to contact us in the future, they are most welcome to Rucha or through other ways. Thank you very much.
Hello.
Yes.
Yes. We have some questions from the participants. Shall I go with the questions, Dr. Itai?
Yes, please. Whatever. We'll be happy to try and answer.
What are the applications of this technique, remote sensing technique, in trapping insect pest attack infestation? Can we predict that, or can we have some application for insect pest attack infestation?
For pest infestation, we talk about like insects.
Yes.
I guess. I've been working on leaf level, which we can detect and see early damage of spider mites. But I think this is not practical for real fields. And it's pending the pest, because some pests it makes sense to use, but it might be that for others it might be too late. I know that people are using and companies are selling services for working with UAV that will take high resolution RGB imagery, and that you can actually see the insects. And then you can identify them and see if this insect is dangerous for this crop or not. But I think I know that companies are selling this kind of service, and I suggest to check with them and see. But if we look at satellites, satellite will cover a big area, and if the whole area is damaged by the insect, I think it will be maybe too late in some cases. Another thing that remote sensing, or not exactly remote sensing, but maybe more GIS can be in service, is that if there are traps for all kind of pests, and you find like the yellow stickers, you can spread them around in, like, I'm talking big areas, and somebody is collecting and checking, and then you can put on the map a point where this is started, where you see them the first time, where you see this for the first time, and then you'll see it on the map, and you can see a pattern of moving and for each direction, and then you can learn from this how to be prepared. And if you know that it's going by wind towards your area, so you know in a week or two it will get to you, so you can be prepared for that. I think this is an option that can work.
Another question is, how do you manage with mixed pictures in the context of spectral images?
Mixed pixels?
Yeah.
Oh,
because when we talk about Indian agriculture, suppose, so we have like very small land holding, and because of that, there are very different crops in the nearby proximity. So maybe that may not be the case in Israel, but still, if you have some other quantities in the same pixel, then how you differentiate that?
I understand. I think that mixed pixels, it's a very challenging issue.
Yes.
Because if you're working, let's say, on a card, like electrical cards that you look for electrical connections, it's easy to work with mixed pixels because you know what to expect. And you can, and you know, I have it or don't have it, and that's it. But if you work in agriculture in open fields to do mixed pixels, it must be with a lot of prior knowledge to train the models. A lot of prior knowledge. I think it's hard to do. What we are doing when we work, we try to work. If you work on the breeding on the small plots, we cut the border. We stay with the heart of each plot. And if we work on fields, we also cut borders. We look, and what we look at homogeneous. We try to get, there is never a homogeneous pixel, but we try to get that. Because I think that unmixing is very interesting topic. It's very complicated, but to apply it in areas that you don't have prior knowledge about, it's challenging.
Yes. Another question is how to use remote sensing techniques in grape vineyards to identify nutrient deficiency. Because in Israel also there are so many vineyards. So how you identify nutrient deficiency?
Nutrient deficiencies. Okay. I haven't worked with vineyards yet, but from what my experience with nutrients is that with vineyards, I would have used UAVs because you can be specific later because you can irrigate per line and you can fertilize probably per line. And I would have used UAVs, and there are several ways to do it. First of all, you need to calibrate a lot. It means like you need to take destructive samples, take leaves, take them to the lab, get the nutrient quantity, whatever you want, micro or macro. But before you do that, you need to have the UAV flying in the vineyard, then select, then connect the spectral data to the plant that you've taken the sample from, and then use it. I know people also working because vineyards, it's important also to have some little stress for the plant to have better yield for wine especially. So this is something that people are using thermal cameras. But for nutrient deficiencies, there are many ways. We've done also models for other plants, but it has to be specific and it has to be with a lot of training.
Okay. And the last and very important question is how you disseminate the research, what you are doing in the lab or the field to the farmers for the proper application in the fields.
Okay. First of all, when we go out, we talk with people and some people talk with us. So sometimes once in a while we get phone calls or emails from people saying, oh, I have this and that, and we talk about it. I cannot say that everything we help, but people sometimes ask questions and they get answers that helps them. On the other hand, we talked about the Chickpea project. We are now in 2 days, we will apply a proposal to get funding to develop the tool that will be available for the farmer in Israel at least. So we will develop some kind, not exactly application, but an internet site that will implement our models for leaf water potential and leaf area index. So a chickpea farmer in Israel can have his estimation of the leaf water potential in the field based on the last satellite imagery acquired.
Okay.
And this will be available free for farmers. We hope to win the proposal and to do that.
Okay. So I think that is all for the questions, Dr. Itay. Shall we close the session now? And at the end of this session particularly, I would like to thank Dr. Itay Harman. He has given a very nice information because we all know about the Indian scenario, but there is very less chance to interact with the people from other countries. What is the scenario in the world? And as per participants' feedback, we are getting very good feedbacks that they also come to know what is the things or technology going on in the other parts of the world. And we are very much thankful to you, Dr. Itay Harman, for sparing your valuable time and sharing the information and your work. Is there any problem? Am I audible, Dr.?
It's with me? I'm
Yeah.
sorry.
Yeah.
No, I'm good. I just want to say thank you very much for the opportunity to present. I must have, like, it was a bit noisy on my end, so I'm not sure exactly if I'm answering to the right question also.
No, that is quite okay. In the YouTube, there is not a problem for the participants.
Yeah. So thank you very much, and I'm looking forward for our own presentation.
Thank you. Thank you. Thank you.
Thank you.
So we'll start with our next session. Our speaker for the next session, he's with us. Arun, are you there? Yeah. So our speaker for the next session is Dr. Arun PV. He's assistant professor in computer science, and he is going to talk about machine learning advancements for smart farming. I'll take an opportunity to introduce him before his expert talk. Dr. Arun is a machine learning researcher and an academician who currently focuses on novel interpretable deep learning architectures for processing and analyzing multimodal Earth observation data. Arun has completed his PhD from Indian Institute of Technology, Bombay. Currently, he is a faculty at the Computer Science Group at Indian Institute of Information Technology, Chittoor. Prior to joining IIIT, he was involved in postdoctoral research at the Ben Gurion University of the Negev, Israel. He also served as a research scientist at Hawaii R&D. His areas of interest include computer vision, deep learning, machine learning, explainable AI, artificial intelligence, multimodal remote sensing, and precision agriculture. He has several papers in reputed international journals and conferences, and is also a reviewer of reputed journals. We are very much thankful to Arun for accepting our invitation, and we are eager to hear you. Should I share the screen, or are you able to share your screen?
Thank you for the introduction, ma'am, and the kind invitation, and for the opportunity. Okay, I will try to share the screen.
Okay.
If any problem, then you can share the screen, and I will join.
Sure.
Okay, is it fine? Is my screen visible? My PPT visible?
Yeah, it's visible.
Okay. Okay, thanks to all of you who are listening me, and thanks for the coordinators for giving me this opportunity. Okay, so the topic here is the role of machine learning in smart farming. So I owe these research credits to Professor Arnon Karneli of Ben Gurion University of the Negev, Professor B. Krishnamohan and Professor Alok Porwal of IIT Bombay, and Dr. Itai Herman, who was the previous speaker. So we all work together. So now going through this presentation, so I will first introduce the context of machine learning to smart farming, and then we will see an overview of the machine learning algorithms, a brief of the artificial neural networks. Then I will try to actually move into, it's with this time constraint, it's not possible to introduce all the machine learning advancements, but we'll focus on one, that's deep learning, or the convolution neural network, which is the jargon of the day, and we will see some applications of convolution neural network to agriculture, and we'll see some research directions, especially the ones that we are focusing on currently. So like all the previous speakers, because I'm mostly from a computer science background, and so you may be aware, like I'm not a domain expert in agriculture, but I was able to collaborate with good people, and so more or less, smart farming is to make agriculture more efficient by using advanced technologies. So we know that, like all the previous speakers were saying, so we have remote sensing technology, we have machine learning. So by this, through these technologies, we are trying to actually improve the agriculture scenario in our country. Like we have just seen, although we have a lot of area being cultivated in Chickpea, in Itai's presentation, but we are not able to get our produce share. So actually smart agriculture is the need of the day. And the basic concept, as you all know, is that every plant should be treated by determining the need of the plant instead of dealing with every farm and crop in the same way. So we should cater to the specific needs of the plant. So this is where machine learning comes into picture. So when we talk about machine learning, so we can say like so through machine learning and this coupled with this remote sensing technologies, we can actually pamper or we can care each plant. So may not be we are we are yet to reach that level, but to some extent we can achieve that. So this talk actually discusses the remote sensing and deep learning approaches to smart farming. So as you all know, and maybe like all the speakers were discussing, okay, this is just a recap of the remote sensing. So we know that we have the source of data, right? So usually in passive remote sensing, so we have the sun as the source of the data, and the satellite collects these reflected radiations, right? And we are getting it in the form of images. So already many people may have covered about the resolution, right? So the spatial resolution is something like, so how well we can distinguish an object on the ground, like spatially, right? So if suppose I say we have like 0.6 meter spatial resolution, 0.6 meter cross 0.6 meter area on the ground will correspond to a pixel. That means the objects which are bigger than that area can be distinguished well, right? So panchromatic sensors will have a pretty good spatial resolution. But we also have multispectral sensors where like we see in RGB colors, but these sensors can see in more wavelength spectral bands, right? So we also have hyperspectral, where actually we have a very high spectral resolution. But the main problem here is there is a trade-off between spatial and spectral resolution. As some of you may have rightly said in the previous lecture, there is a spectral mixing problem, right? So why is it so? Because as we increase the number of spectral bands, the energy that we are getting from the Earth's surface actually decreases. And so we have to focus on a large area. So that way, actually, the spatial resolution decreases. So there is a trade-off between these two. So it actually depends on what is our application. What actually we want to do. So accordingly, we have to select the sensors. Sometimes we can use multiple sensors. We can fuse together multiple sensors. So there are a lot of applications. Many of which have been already discussed. So this is a satellite image of an agriculture field. Like this is in different, different bands we are giving importance to, so we can have, we can have a different views. So, the spectral signature of a crop. So when we say the pixel spectra, if we have the satellite image, so actually each pixel, like if suppose it's a hyperspectral satellite image, right? So we have like 225 spectral bands. So each pixel will have a length of 225. It will be a vector of 225, right? So we can say if we plot it, so we will get something like this, a spectral signature we will get. So this pixel vector or this signature, spectral signature, we can check it to detect the vegetation type or anomaly. We can compare it with already existing libraries to see what type of vegetation is there, and if it is deviating from that particular spectral signature, right? So this is another like healthy or stressed sugar beets. This can be also detected. So another way, like right now, what I was saying was like, see, we can detect things based on the spectral bands, right? So this is for a, single instance. Given a particular instance, I will just observe the plant, right? What is its difference? So I will just, try to classify it based on that, right? So, but if we have time series data, sequence of data, because always a particular instance, maybe two crops will look similar, right? But if we have a time series data, then we can have like further distinguish this, because within a particular season, some crops may behave differently than the others. So this is where the time series data comes into picture. So we can have different, here we have plotted the NIR reflectance, but you can also have different type of indices, crop indices also to, to plot such a signature or such a curve, and we can classify crops based on such curves. So this is when we have time series data, and the other one is when we have only a single instance image, maybe hyperspectral, multispectral images. So this is again another example of the same thing. So we can identify the vegetation, right? In one image we can see we have the, so here we can see we have this actual image, and here we can see we have given some labels, right? Labels to these pixels, so that actually we can have a better understanding of these different categories of vegetation. So here again, vegetation health monitoring. So given an image, you can have, given a satellite image, you can have this type of an image using some techniques, right? So where each of the pixels is labeled based on the, on a set of labels, like whether it is healthy, whether that crops are fine, whether it is under stress, right? So what is the role of machine learning in doing all these things? So we have seen, like, okay, we have remote sensing data, right? We can do it. So machine learning algorithm is something which, based on the data, we should be able to predict something. So how this is possible? This is like, I'll give you a very simple analogy. All of you may be aware, but so just to recap it, and when we were kids, okay, when we were small kids, so how we learn to distinguish different fruits. So for example, okay, someone will bring us an orange and will say, right, okay, so this is orange. So first we may not capture it, but upon seeing different oranges, then we will see like, oh, this is orange. So we slowly learn, okay, this is orange. So someone will give us a feedback, right? For example, when we say apple also, at first we will say, okay, this is also orange. Then our mother will say, no, beta, this is not orange, this is apple. So we will correct ourselves, right? So we actually generalize in our mind what is the concept of orange. We understand using our senses. These are the parameters, right? So an orange in our brain, it will be mapped based on those parameters, maybe the color, maybe the shape. These can be the parameters for distinguishing an orange from other fruits, right? So maybe the smell, different parameters. So we generalize this. So same way, machine learning algorithms, we give them first the input data and what is required, right? Input data means we will show orange and we want it to say it's orange or label it as orange, right? So first when we give this data, it will, it may wrongly predict. Then we will give a feedback. Okay, then it will correct that error, and that is how machine learning algorithm learns, right? There are different approaches, to generalize these rules or these concepts. So for example, how to label a particular pixel in a satellite image. One way is that when I say label, means assign a particular group or category to each of these satellite image pixels, right? So we get a satellite image. So what we actually do, we assign a label to each of these satellite image pixels. How we can do that? One way to do is to manually check each pixel and assign a label, right? For example, the crop type. I'm given an image. I will check each pixel. I will check each pixel. You should remember, if it's a hyperspectral image, it says pixel spectra, right? I can compare it to the library spectra, and I say, oh, okay, these are matching, so these belong to this crop. Another way is to use a machine learning algorithm to automatically assign the label or value, right? So we have a machine learning algorithm. Here you can see we have some spots, right? These are the training data which I have used. So I use this training data to tell the system, okay, this means this is for us, this means, if this value comes, this means this is something else. So that way I will tell the system. And you can see wherever there are no spots that were once I trained, that were classified by the algorithm by itself. So this is a recap of machine learning. So we can say, like, pixels are labeled to generate classification maps, right? Crop type maps, crop health maps, like we have just seen. Also, we can regress them to generate yield maps, moisture estimates, etc. So, you hear about a lot of machine learning algorithms and approaches, right? So, usually to just simply categorize these different things, I'm just presenting a very simple, a brief categorization of this machine learning algorithm. So if we want to predict a category or a class, right? So we use, if we have label data, then we go for classification. If we don't have label data, we go for clustering. And if suppose we are not predicting a category but a quantity, right? So we go for regression. And if we are trying to select the best representation from the data, so we can go for something called dimensionality reduction. So there are a lot of approaches which comes under the umbrella of classification, umbrella of clustering, dimensionality reduction, regression, these things. And there can be many other things as well. Okay. So when we say, okay, image classification, like I discussed briefly earlier, we will make our system to generalize, right? While I gave some of these samples, my system was able to generalize, right? So what is exactly happening here? When I say the training phase, what is happening is the system will learn some subspaces, and it will see these subspaces based on, say, here band 1 and band 2. So you have to imagine that the system sees the image in this way, like a scatter plot, not the way like we are seeing like an image. So here, in many cases, the spatial information may not be considered. Only the spectral information may be considered, or the value of the pixels may be considered. In this case, only the value of pixels are being considered, right? So we have sub-regions, and we try to put that pixel. The system will, when a new pixel will come or a new value will come, which we want to classify, right? So basically, from the data, it has already learned that, okay, if it falls here, this will be this class, this will be this class, this will be another class, right? So based on where it falls, we can decide that class or category, right? So this is basics of this classification algorithms. And so we will specifically focus on the neural networks. So ANN, as you all know, ANNs are computational models that mimic biological neurons. So a weighted sum, each node in a neural network computes the weighted sum of the input data, which is squashed using some activation functions to yield the output. So what exactly are we going to do with the neural network for classifying a remote sensing image? So when we want to classify a remote sensing image, we have a neural network, right? So data is presented to the network in the form of activations in the input layer, right? So each spectral band is a variable, right? And error is propagated backwards to train the network. For example, we have, suppose we have 1 pixel, right? This is 1 pixel. So 1 pixel means we have a curve like this, right? We have a vector value like this. So this value we will be presenting to our network one by one. I mean, first, maybe the red value we are presenting to the network, right? There is a set of hidden layers. So like I just discussed, so this will aggregate all the incoming nodes, right? And we will get in the output layer some labels, right? So based on this, we can have, I mean, instead of this image, we can have a labeled output image. So this is how actually a neural network algorithm works. This is a very surface, I mean, discussion. I'm not going very deep into the mathematics or how this error is being propagated. So there's something called back propagation. I think most of you who works with machine learning may be aware, and we have to optimize that weights, right? I mean, similar in case of regression, we adjust the coefficients. So same way here, we are adjusting the weights, this network weights. So that way we will make the network to finally learn something. So when we say, okay, network learns something or it's able to predict predict a given class or classify something, what actually the algorithm is doing is that it's learning a decision surface. I mean, same band 1, band 2 in the feature space that I have shown earlier, right? So it's a linear decision boundary. So we are trying to the network, once it is trained, the network try to construct a decision boundary between the classes. So this is through error propagation that it happens. So you can see here, like you can see first the decision boundary was somewhere else, right? So upon training, it slowly comes and fits into that place through the optimization procedure. One example is the back propagation algorithm. So when we talk about the decision boundaries of, I'm just talking about these things because we want to have a motivation of deep learning, right? So why we have to go deep? So basically what we see here is that perceptron or a single layer. When we have single layer, we can have a linear boundary like I have just shown here. When we have MLP, multi-layer perceptrons, we can go more complex decision surfaces. So I mean, as you go more, I mean, depth equal to 1, you can see here depth equal to 2. So as you go more and more deeper, we get more and more complex surfaces, decision surfaces. So you can see here, more complex surface is required to classify this particular distribution, right? Because a simple linear surface, you cannot classify. This belongs to one class and this belongs to another class. Our aim is to find a decision surface to separate these classes. But here it's not possible. It's here somewhat possible. Here it was I mean, better, it could classify it in a better way, right? So this is how slowly the decision surfaces are being modeled. I mean, by using, I mean, how we can correlate the decision surface with respect to the depth of the network. So we can say in one way, okay, deeper means better. So what exactly is deep learning? Is simply increasing the number of layers deep learning, or is there some intuition behind it? What actually differentiates machine learning from deep learning? So in machine learning actually means we are, when we give an input, right, we extract the features and then we give it to the system, and system classify or do something. I mean the neural network or the machine learning model do something based on those features, right, and we get some output. But in deep learning, we do this feature learning and this classification or whatever be our objective both together. Okay? So I think so in that sense we can better fine tune this whole procedure. So it has been found that these deep learned features are much better than this. I mean, than these handcrafted features. Handcrafted features means the features that we design, right? For example, sometimes if you are working with agriculture crops, okay, you get a spectral reflectance, right? So you give that spectral reflectance to the system. I mean, where each feature means features are the, I mean, the spectral response for each wavelength, right? So maybe one way ahead, you can extract some specific wavelengths and give to the system, right? So that these are all manually designed features. So what if the system itself can see which feature is more useful, right? So that is where the deep learning comes into play. So we don't have to specify much things. The system will try to find out which features are more good for a particular objective. So autoencoder is a type of neural network where we give the input and we get the same input as output. So what is happening here? What is the, you may ask me, okay, why we are doing so? Because we want to learn the compressed representation of this data. So this will be the learned representation. Right? So what we say is we can have a better manifold or a better representation. This representation is always called latent representation of the data. So a learned feature space. So when I say a learned feature space, may sometimes, if this is the original image, so first level features may correspond to edges, right? The next level features may correspond to object parts, and at the end we will get the object. So basically, to classify this image as a face, the system will learn all these hierarchical features, right? But there is a problem with the fully connected network, that and fully connected network means every node is connected to every other node, right? So there we have a lot of trainable parameters. So to deal with it, this is a fully connected network. So you can say every node is connected to every other node, right? So to deal with it, we came up with convolutional networks. So I mean, this was proposed long back by Hinton. Okay, so this convolutional networks, but they are not connected to all the higher level neurons, right? But only to certain neurons which are in the vicinity. I mean, this neuron is only connected to this and this neuron. Same way, this is connected only to these three neurons. So there is one more, is the only advantage of convolution neural network that we don't have a lot of weights. There is also another advantage, that some image features, right? Like last time I told, we cannot consider the spatial nature or the spatial context. Okay, so by modeling these connections in a particular way, we can actually make the network to realize the context. I mean, what context is your pixel in? I mean, earlier we were only using one pixel to classify, to label it, right? So now we can also use the neighboring pixels as well. So this will give you more accuracy. So this was the case if we give an input image. If it was an ordinary neuron, this entire pixels would have been connected to this neuron. This entire pixels would have been again connected to this one, etc. Right. So now, in a convolution neural network, only certain context will be connected to, certain neighborhood will be connected to one neuron, right? So we have significantly reduced the number of connections. And not only that, we can also learn the context now. I mean, local information. So by having additional layers, we can actually model all the local information. Then we can combine this local information to get a global information. So this is how a convolutional neural network will look like. So you can see here, if this is the input image, right? X1, X2, X3, X4, X5 up to X time. Right? So we can connect it in such a way that, so this kernel, when I'm placing over here and moving in, so this process is called convolution. So we can simulate that convolution by connecting it in a particular manner. So it will be more clear now. So what convolution actually means is, like, we have an image, right? So when I told you about, I mean, the features being learned by the system, right? So how it is being learned? So this is one feature derived from this image. So when we use this particular filter, what we do here, we move this filter over this image by a weighted multiplication of each pixel value by each of these filter coefficients. So we will get something like this. If we use another filter, we will get another feature. So this process is called convolution. So to make it more clear, what we are doing here is, here our filter is this one, 1 0 1 0 1 0 1 0 1, right? So it starts moving across this image, and we are getting a new feature here, right? Here multiply and sum it, so we are getting convolve features. So this is same. I mean, when you are doing this filter over this image, we will get this one. This is a mathematical representation of the same thing, right? So when we get a large image, say 6 by 6 image, say we have the filter 1 and we have filter 2, right? So we put this filter over this one, right? So what we do, we multiply each of the filter coefficients with this image image values, right? Corresponding image values, and we add it like 1 into 1, minus 1 into 0, minus 1 into 0, minus 1 into 0, 1 into 1, plus we add all these things together, we get the value 3. So then it moves, we get another value, right? So like this we are getting. So this process is called convolution, right? So again, if we have another filter, like here filter got changed, now we have another filter. So same thing we do for that filter also. So we get another feature, right? So all these features together, feature 1, feature 2, all these features are called feature maps. So these will be the learned features, right? So actually, why I showed you this convolution is that the same operation we can implement using neural network. How it is possible? So here you can see I have here we have given different colors, right? So this is the filter, right? So what is happening here? So if you consider this particular window, say the first window 1 0 0 0 1 0 0 0 1, right? So we are connecting all these things. So this is in case of a fully connected network. But sorry, this is in case of this convolution network. So we are connecting all these things in this manner so that we can say like this 3 is resulting from the convolution of this filter 1 over this particular image. So same way, this is the next one. When you put it over here, so you can say like this one, this one, this one, right? So for example, the second one, the third one, fourth one. Now, see the fifth one is not connected here. The sixth one is not connected here, right? The seventh one is not connected. Eighth one is now connected, right? So the eighth one is connected here. The ninth one is connected. The 10th one is connected. This is the 10th one, right? So now the 11th one is not connected. The 12th one is not connected. The other one will be connected. The 13th one is not connected. The 14th one is connected. So this way we can say like we can implement the convolution using this neural network setting where we have two advantages. We capture the neighborhood information as well as we have fewer parameters. So this is why this framework or this model is called convolutional neural network, because this can better be used in our for images. So what we actually get now is that we got the features, right? So I told you each feature will represent, one layer means it will represent one context, right? We not only need the local context, but we also need the global context. So how you can move from this context to higher is by using more and more layers, right? So in between, when we go from one layer to another, we use something called a max pooling layer, so that we try to reduce the resolution of these features, so that at the end we can get very less information, but which hold all those global features. So like we can have different type of pooling, and so at the end we will just flatten all those. Once we have like say convolution layer followed by pooling layer, then again convolution layer, again pooling layer, like this we go in. At the end we will, if we have given a big image, right, we will get a very few set of features representing that entire image. So now we can just flatten them and we can use them for classification, right? So using a fully connected network. So we have learned these features. These are the features that we learned. These features will represent that entire image better than directly giving that image to this neural network, right? So when I was explaining it part by part, we have a convolution. We have an input image, right? So we have a convolution unit. It will give you a feature. Then we go to max pooling, right, to reduce that resolution and to pick out only certain parts so that we can go to a higher level. Then again convolution, then again max pooling. So here we are getting the features. These features are getting flattened and is fed to a neural network to classify. This neural network will say whether it is a cat, dog, etc. Right. So what happens in case of our crop spectral signature? So instead of an image, we can give that spectra to this one, and it can classify it as wheat, rice, or something. Okay, so these are some of the examples. So as I already explained to you, so at different levels, we can have different features, right? So that is the beauty of convolution neural network. And so there are different examples for this one. So we can say like the different levels, we learn different things. The same network, you can see it learns, different features. It gives different importance to different features for different objects so that we can easily distinguish them. So it can actually model the spatial context, can learn the hierarchical features, less number of hyperparameters. And also one more thing is like in one lecture, the speaker was saying like, okay, there is a problem with the machine learning models that if you train on one area, we may not be able to train on another area, right? So with deep neural networks, there is something called transfer learning, which to some extent can resolve this issue. So when we talk about this, just this classification, so what we are actually doing, we are here learning the features, right? And we are using a classifier such as neural network to predict the label. Or another way of doing it is we can use an encoder decoder to again reconstruct the spectra. But these features, we can classify it using any classifier. Say example SVM also, support vector machine also. So in this case, it's an end-to-end system. You have to use the classifier, the neural network based classifier. But here you can just learn the features and you can use these features. You can classify it using any classifier. So we can also learn the spatial context. Okay. Using CNN. So it's very obvious that here instead of the single pixel spectra, like in the previous case, we also take here the context, this 3x3 or 5x5 neighborhood, and do the same procedure. So we can have a lot of classification using the CNN. And why I'm stressing on this deep learning and convolution networks is that because a lot of recent research have reported state-of-the-art accuracies using these approaches. But like I also told, it's not that we always have to use deep learning. It also depends on the circumstances, how much data you have, and what is the type of the problem you have. Sometimes very simple classifiers such as SVM or even K nearest neighbor can give you better results than deep learning. So we have to think about what exactly we want to do. Based on that, we have to decide the model. And even the length of, depth of the deep learning model, all these things, the number of filters that you have to use, all depends on the particular application. So again, the chlorophyll, LA estimation, all these things we can use these networks. So another area is the dimensionality reduction, which is very frequent in case of agriculture research or related researches, where we have a lot of variables and we may have to select a few of them, right? So another way is like to transform these 100 to another set of variables where majority of the data or the information is contained in the top variables, right? So principal compound analysis is one example. So instead of using PCA or principal compound analysis, we can also use encoder decoder type of approaches just to reduce the dimension. And so instead of using these variables, we can use these two variables, right? So we can easily get a low dimensional representation. So many studies have shown that the autoencoders can better represent the data. But it also again depends on different applications and your data. Okay? So it's not, not an absolute statement that always this can perform better. So I'm just saying another application of CNN is the segmentation part, where we can actually have we can segment and we can given an image, we can segment and we can label it. So this also finds a lot of application where we have a satellite image, or even we have a close range photograph and we want to categorize, or count how many seeds are there, or even this is applicable for close range photography of a plant, right? So where we have to segment it, we need to find the root length or the shoot length. All this we can use a CNN based approach and we can segment it, and then we can measure those measurements. CNN based weed detection. So there are different approaches. This is a very simple framework that I presented before you. So there are a lot of advancements based on that. Okay. So object detection, basically, it's very applicable in case of smart agriculture, where we can count the number of fruits, or even we can, yield estimation, and we can check actually, if there is an infection there, right? So different applications are there for the CNNs, and the weed detection is another area. So to summarize, actually, we have a lot of machine learning algorithms, and we can say like the state of the art results are being reported from these deep learning techniques. So this I will just discuss the last 2 points. I'll just discuss in the coming 2 slides. So this is about our recent research directions which is going on. I won't be going into the technical details. I'll just explain the problem what we are actually doing. So someone was asking about spectral mixing. So this is a problem that we all face. So how to solve it using convolutional neural network is one area that we are working on. There are a lot of papers, a lot of algorithms that you can actually see. So what they basically do is there is something called spectral unmixing and the subpixel mapping, where we try to find the n members and we try to unmix. The unmixing means actually the abundance contained in a particular pixel, right? And the subpixel mapping means where is it located. So actually spectral unmixing means we actually get only one value, right? From this value, we need to identify, okay, how much of material 1 and material 2 are there, right? So this is spectral unmixing. Okay. And another area is the spatial resolution enhancement, right? Given an image, and if you remember, this is almost the same image that Kai has shown you. So a similar image. So if we have a core spatial resolution there, how can you improve the spatial resolution of these remote sensing images? So that is another area. Not only the spatial resolution. For example, suppose you have a multispectral image, can you improve or can you use some pre-trained some model, or using some sort of data which already existed, some learning the correspondence between some data to improve its spectral resolution. Okay, this is also something which we are experimenting. And another very important part is this feature modeling. Like for example, if when I'm giving something to a neural network, okay, it can say, okay, classified this into this class, right, into another crop. If I'm giving this spectra, it can classify it into different crop classes. But what it is actually focusing on, is it giving importance to the features which actually distinguish that 2 classes? So that is another area that we are looking into. So that is again another area is this multi-model. Like I told you, one data may not be sufficient. So how you can fuse together all the different data to get mapping in a better way. Another very important part which I just covered again is this because network, when I say this neural network, okay, it is learning these features, right? It's again a black box, what features it is learning and how we can explain that model. Suppose my model is properly classifying wheat and barley, right? So what that model is focusing on, what features it is focusing on, are it focusing on, is it focusing on meaningful features or not? So these things comes under this interpretable convolution network, where we try to interpret the network or try to explain the network's behavior. So these are some of the areas that you can also try. So that is all from my side. Thank you again for the kind attention. The aim of this talk was basically to introduce you to the jargons in the machine learning, and especially to deep learning and the convolution networks, so that if any of you are interested, at least you will get an awareness of these things are there and you can use it for these purposes, and it's not that difficult to understand them. Okay, so that was the aim of this talk. Thank you again for your kind attention. I'll be happy to take questions.
Thank you, Arun, very much, because we are getting very good questions from the participants also. So should I go with the questions?
Yeah, sure.
Yeah, one of the participants, he has asked, why is CNN mostly used in classification processes rather than ANN in plant management?
Yeah, so like I told initially, so what actually happens here is like we, I mean, what was the area that he was referring to?
Plant science, plant management.
Okay, plant science. So basically, if we want to understand the context, right? So it's better to use the convolution networks. Like you have seen, I mean, simple neural network may not always count the context, right? So because the local context and these features, I mean, hierarchical learning of features is which make convolution neural networks perform better than the normal neural networks. So what we can see here is like by learning, by not having a fully connected network, by having these segmented connections or shared weight connections, we are actually learning a lot of features which actually make a lot of physical relevance. So that is why increasingly in images and other areas, we are more using these convolution neural network, because those features will be more meaningful, and more than that, actually this is learning the spatial context, taking the spatial context. In neural network also you can incorporate the spatial context, because you can have a texture image, right, and you can give the texture image as input to the neural network. But there again you have to remember it's handcrafted feature. So here the features are automatically learned by the system. So that is one.
Another question is, which software is good for machine learning? Is our software sufficient, or we have to go with the Python or any other software? Am I audible? Arun, am I audible? Is there any problem from your side?
Okay, am I audible, ma'am?
Yeah, no.
Yeah, okay. So sorry, there was some network problem, technical problem.
Okay. So which software is good for machine learning? Is our software sufficient, or we have to go with Python or any other software?
I mean, so far I think R looks good if you are doing some preliminary type of analysis or something. But if you want to design your own networks or something, then I would advise you to go ahead with Python. I mean, it all depends on how much depth we want to go in. I'm not saying like without, I mean, with R you cannot go in much depth, but I mean, in Python there are a lot of developments happening and a lot of libraries, lot of packages coming in, because all these high-tech firms are working in actually Python. So it gives you a more more resources. It will give you more resources actually. So yeah, so it actually depends on what particular application or how deep we want to go.
I think that is all. There were 2 questions from the participants.
Okay. Yeah.
Thank you, Arun. Thank you very much for spending a very quality time with us, with your knowledge and whatever research you have done. We are very much thankful. From Anand Agricultural University, I thank you. And at the end of this webinar, I request Dr. Manjusha Kulshrestha, Head of the Department of Basic Sciences, to convey vote of thanks to all the speakers and for the entire webinar. Please.
Thank you very much, Rucha. And I think here is a problem of internet connection. So maybe everybody will find difficulty. But even though I'll go on my thoughts or showing my sincere thanks on behalf of our team of Platinum Jubilee Celebration, BACA and NAHEP Cast. On behalf of them, I'll sincerely give my gratitude to our patrons, Dr. KB Kathiria, Honorable Vice Chancellor, Anand Agricultural University, cooperating and showing their interest on organizing this webinar. Also thankful to Dr. R.C. Agrawal, DDG Education, National NAHEP, ICR, New Delhi. Also thankful to the advisor of this event, Dr. Prabhat Kumar, National Coordinator, NAHEP, Cast, ICR, New Delhi, and Dr. M.K. Jala, Director of Research and Dean PG Studies, cooperating to organizing such this good and good theme seminar. Also, this is my sincerely, I appreciate the means from the team that the keynote speakers, without them, it's keynote speakers and guest speakers, which will not be become fruitful and helpful to the students and faculty members. And this such type of topic is very new and very useful for this such a this need. It is a need of the time. The keynote speaker, we are very thankful to Dr. Bimal Bhattacharya, who is Group Director of BPSG, SEC, ISRO, Ahmedabad. Guest speaker, we are very thankful Dr. Itai Harman, who has cooperated us for the, even though he has different time zone from Israel. And thankful to the another speakers, Dr. Prashant Srivastav, giving the good information on this ML and AI and with this applications in precision farming, who is from this BHU, India, Dr. Arun PV from IIT Chittoor. Thankful to all these speakers and keynote speakers, and simultaneously we are also thankful to the conveners of this webinar, Dr. Vayam Shukla, who is Principal and Dean Baka and Agriculture Faculty, and also convener of the Platinum Jubilee Celebration Baka, Dr. R.S. Pundir, Principal and Dean IABMI, and Principal Investigator of NAHEP Cast, AAU Anand. Also, without fail, I should show my thanks to Rucha Dave, who is co-PI of NAHEP Cast, and nicely organizing this webinar and conducted a very good way. And also it is became effective because of her. Thankful to Dr. R.G. Parmar, Organizing Secretary, who is Platinum Jubilee Celebration Baka, and co-organizing secretaries Dr. Y. Lard, who is CCPI NAHEP Cast, and Dr. D.B. Sisodia, co-organizing secretary, Platinum Jubilee Celebration Baka. So once again thankful to each and everybody, and also the important is the audience and students and faculties, those who have participated. Without them, this webinar cannot become fruitful. So I sincerely appreciate your attention today. Thank you very much.
Thank you, ma'am. And now we shall call off this webinar and have a nice time ahead. Thanks to all.